7 minutes. That is how long an extinct species stayed alive after we brought it back. One goat in Spain in 2003 before its lungs failed and it became the only animal in history to go extinct twice. Right now, there are 3 wolves living in America that shouldn't exist. These are the 10 animals coming back, ranked from furthest away to closest. Number 10, the passenger pigeon. Start with a number that's hard to hold in your head. There were somewhere around 3 to 5 billion passenger pigeons in North America. Not millions, billions. They were possibly the most abundant bird on Earth, and flocks were described as darkening the sky for hours at a time. A single flock could take most of a day to pass overhead. They were gone in about 50 years. Commercial hunting, telegraph lines to report flock locations, railroads to ship the bodies to cities. Industrial slaughter on a scale that nobody thought could dent a population that size. The last wild bird was shot around 1901. The last passenger pigeon alive was a female named Martha, and she died in the Cincinnati Zoo on September 1st, 1914. Her body was frozen into a block of ice and shipped to the Smithsonian, where she still is. The revival effort is run by a group called Revive and Restore, and it's been going since 2012. 14 years. There is no living bird. Here's why it's last on this list, and it's not just the technical problem, though that's real, and we'll get to it with the dodo. It's the behavioral one. The passenger pigeon's entire biology was built around living in flocks of hundreds of thousands. They nested communally. They fed communally. They defended themselves by sheer overwhelming numbers. There's good evidence they simply couldn't function in small groups, which is part of why the collapse, once it started, was unstoppable. So even a technically perfect passenger pigeon would be a bird without the one thing that made it a passenger pigeon. You'd have the animal and not the species. You'd need thousands of them at once to get anything resembling the real thing. That's the first version of a problem you're going to see over and over as we climb this list, and it gets worse the closer we get to the top. Number 9, the Aurochs. This one belongs on the list for a completely different reason. It is the only project here that does not use modern genetics at all. The Aurochs was the wild ancestor of every cow alive today. Enormous. Bulls stood up to 6 feet at the shoulder, with forward curving horns that could reach 3 feet. They ranged across Europe, Asia, and North Africa, and they are the animal painted on the walls at Lascaux. The last one died in a Polish forest in 1627, which makes it one of the best documented extinctions in European history. The method being used to bring it back is essentially the opposite of everything else in this video. It is called backbreeding. The idea is that because the Aurochs genes never fully disappeared, they are scattered across primitive cattle breeds all over Europe, and you can concentrate them again by selectively breeding the most Aurochs like animals over generations. No CRISPR, no cloning, just cattle and patience and a lot of time. There's a dark chapter here too. The first serious attempt was made in Nazi Germany in the 1920s and 30s by the Heck brothers, 2 zoo directors working within an ideology obsessed with racial purity and ancient Germanic landscapes. They produced Heck cattle, which still exist. Most scientists consider them a failure. They look vaguely Aurochs-ish, and genetically, they're not close. Modern European projects are doing it properly with genomic guidance. They've produced animals that look strikingly like the ones in cave paintings. But that's the honest limit of it. You're building an animal that resembles the Aurochs from cattle that carry fragments of Aurochs ancestry. It's a portrait, painted in livestock. Number 8, the Gastric-Brooding Frog. This is the one where you feel the loss most sharply because of what this animal could do. The Gastric-Brooding Frog lived in a small area of Queensland, Australia. The female would lay her eggs and then she would swallow them. All of them. Once they were inside her stomach, she would shut down her own digestive acid completely. Her stomach stopped being a stomach and became a womb. She didn't eat for about 6 weeks. The tadpoles developed inside her, and then she gave birth by opening her mouth and letting fully formed baby frogs climb out. An animal that could switch off its own stomach acid on command. Think about what that could have taught us about human ulcers, about reflux, about gut medicine. It was discovered in 1973. It was extinct by the mid-1980s. We had it for barely a decade, and the likely culprit was chytrid fungus, the disease that's been devastating amphibians worldwide. The revival attempt is called the Lazarus Project, run out of Australia, and it got further than most people realize. Using frozen tissue and cloning techniques, the team produced embryos that started dividing. Early-stage embryos of an extinct animal alive in a dish. Then they stopped dividing and died. That's where the project stands. It's genuinely close in one sense. They've had living cells and stuck in another, because getting past that early developmental barrier has proven extremely hard. Number 7. The giant moa. New Zealand had no native land mammals. What it had instead were birds that grew into the roles mammals fill everywhere else, and the largest of them was the giant moa, standing up to 12 feet tall with its neck extended. Flightless, wingless, an enormous herbivore that had no idea what a predator was, because until humans arrived, there was almost nothing that hunted it. Polynesian settlers reached New Zealand around 1300. The moa were gone within roughly 200 years. It's one of the fastest megafauna extinctions on record, and it's uncomfortably clear-cut, no climate mystery, no debate. People arrived and ate them. The MOA is the newest addition to Colossal's project list, backed in part by Peter Jackson, who is a serious MOA bone collector. And the technical problem is the same one that puts the dodo where it is on this list, the bird problem, which we should actually explain now, because it's the reason two of the most famous extinct animals in the world are stuck. Number 6, the dodo. Everyone knows the dodo. Almost nobody knows why it is so much harder to bring back than a mammal. Quick correction on the animal itself, since it's one of the most misrepresented in history. The dodo was not fat, stupid, or clumsy. That image comes from drawings made of overfed captive birds in Europe. The living animal was a large, capable, flightless pigeon that had evolved on Mauritius with no ground predators. Its fatal flaw wasn't stupidity. It was having no reason to fear anything. Sailors arrived in the 1500s, and the dodo was gone by around 1690. It probably wasn't sailors eating them that did it, which is the other half of the myth. Accounts suggest dodo meat wasn't especially good. What actually killed them was everything that came off the ships alongside the sailors: rats, pigs, cats, and macaques. The dodo nested on the ground and laid a single egg. It had spent its entire evolutionary history on an island where nothing ate eggs. Within a couple of generations of rats arriving, that strategy went from perfectly sound to fatal. It took less than a century. And here's a detail that says a lot about how little we cared at the time. There is no complete dodo skeleton from a single individual anywhere in the world, assembled from one bird's bones, and no preserved soft tissue of any real quality. We had the animal for 100 years and barely bothered to keep any of it. Now, the technical problem. Every mammal in this video can potentially be cloned or edited using the same basic route. You take a cell, you edit the nucleus, you put that nucleus into an egg, you implant it into a surrogate. You cannot do that with a bird. A bird's egg isn't a cell you can inject a nucleus into and implant. The yolk is a single enormous cell. Fertilization and development happen inside a shell that's assembled as the egg travels down the oviduct duct, and there's no womb to implant anything into. The entire mammalian playbook is useless. So the workaround is genuinely clever. Instead of editing an adult cell, you edit primordial germ cells, the cells that will eventually become sperm and eggs. You put edited germ cells into a developing embryo of a related species. That bird grows up as an ordinary bird, except its reproductive cells carry the edited genome. Then, you breed those birds and their offspring are the target animal. For the dodo, the base species is the Nicobar pigeon, its closest living relative. Colossal has completed high-coverage genomes and reported successful gene editing in bird germ cells, real progress on a problem that was considered near impossible a decade ago. But notice what that method requires: an extra generation. You don't get a dodo, you get a pigeon that might produce a dodo-like bird. That's why the birds sit at this end of the list. Number 5. The Pyrenean Ibex. Here's the one that's already happened, and it's the one that tells you the truth about all of this. The Pyrenean Ibex was a wild mountain goat from the Spanish Pyrenees, heavy-horned, built for cliffs. By the 1990s, there were about 10 left. By 1999, 1. Her name was Celia. Scientists caught her, took a skin biopsy, froze the cells, and released her. 10 months later, rangers found her dead under a fallen tree. Not poachers, not disease. A branch came down in a forest in northern Spain, and a species ended. January 6, 2000. But the cells were still in a freezer. A Spanish and French team used the technique that made Dolly the sheep. Strip the genetic material out of goat eggs, insert Celia's DNA, implant the embryos into surrogate goats. The numbers are brutal. Hundreds of reconstructed embryos, only 5 pregnancies, only 1 carried to term. On July 30th, 2003, they delivered a female kid by cesarean section. Normal heartbeat, eyes open, legs kicking. For the first time in human history, a species that had been declared extinct was breathing. She just couldn't breathe well. She'd been born with an extra lobe in her left lung, taking up too much room in her chest and preventing that lung from inflating. Everything else was normal. Her DNA was a clean match to Celia's. No lethal mutations, no other defects. One structural flaw in one organ. After several minutes of severe respiratory distress, she died. 7 minutes, less time than it takes to boil pasta. And the part that makes it worse, even if she lived, it was over. Cloning from Celia's cells could only produce females. There was no male Pyrenean Ibex anywhere on Earth. A perfectly healthy clone would have grown up as the last of her kind all over again. That was 23 years ago, and it has never been repeated. Every project in this video is measured against those 7 minutes, which makes the next one on this list almost unbearable because it's the same story happening right now in slow motion, and we can still see the ending coming. Number 4, the northern white rhino. This one is different from everything else here because it isn't extinct yet. There are 2 left, Najin and her daughter Fatu, both female, both in Kenya, both under 24-hour armed guard. The last male, Sudan, died in 2018. When these 2 are gone, that is the end of a subspecies that numbered in the thousands within living memory. Sit with that for a second, because the scale of it is easy to skip past. This isn't an animal we lost to the Ice Age or to sailors in the 1600s. Northern white rhinos were still being poached in numbers in the 1970s and 1980s. There are people alive today who are older than the entire collapse. And the cause wasn't habitat loss or climate. It was horn, a substance chemically similar to fingernails, sold for money. Neither of the remaining 2 can carry a pregnancy. Najin is too old and has a condition that makes it dangerous. Fatu has a uterine issue. So the line cannot continue naturally under any circumstance, no matter how well they're protected. The armed guards aren't preserving a breeding population. They're guarding the last 2 members of a species that has already functionally ended. What's happening instead is a race. A consortium called BioRescue, which Colossal joined in 2023, has been harvesting eggs from Fatu, fertilizing them with frozen sperm collected from males that are already dead, and creating embryos. Those embryos exist right now in cryogenic storage. The plan is to implant them into southern white rhino surrogates, a closely related subspecies that can still carry a pregnancy. I'd argue this is the most important project on this list, and it gets a fraction of the attention the extinct animals get. There's no debate about whether it counts, no argument about species definitions, no marketing problem. 2 animals are alive right now. The clock is running, and every technique the de-extinction field has developed is being aimed at stopping a line from ending. It's also the clearest demonstration of the thing that makes all of this possible, which we'll come back to at the end. Number 3, the Tasmanian tiger. Of everything on this list, this is the one people genuinely want back, and it has the best case. The Thylacine wasn't a tiger and wasn't a dog. It was a marsupial predator, striped across the lower back, roughly the size of a medium dog, with a jaw that could open to a genuinely startling angle. It lived across Australia and New Guinea before dingoes arrived, and survived in Tasmania until Europeans decided it was killing their sheep and put a bounty on it. Thousands were killed for money. The last known Thylacine died in a Hobart zoo on September 7, 1936. The way it died is the detail that stays with you. His keepers called him Benjamin. He'd been at Beaumaris Zoo for 5 years. On his last night, Hobart had been unusually hot during the day, and then temperatures crashed to freezing after dark, and Benjamin had been locked out of his sheltered sleeping quarters. Some accounts say it was punishment for bad behavior. Others say a keeper simply forgot. Either way, the last known member of the species died of exposure in a concrete enclosure because someone didn't open a door. The species had been granted legal protection 59 days earlier. We have footage of him. There are only about 10 known film clips of living thylacines in existence, and the longest runs roughly 80 seconds, shot by naturalist David Flea in December 1933. You can watch Benjamin pace his enclosure while a keeper rattles the fence trying to provoke one of the species' famous threat yawns, where the jaw opens to an angle that looks anatomically impossible. That's the entire moving record of an animal that once ranged across a continent. Small correction, because I see this constantly. 1936, not the 1980s. The IUCN didn't declare the species extinct until the 1980s, which is where the confusion comes from. But the last animal died in 1936. There's a thread here that refuses to die. People keep reporting sightings. There's a database of over 1,100 reported thylacine sightings, and the reports haven't tapered off with time. In some regions, they've actually increased. In March 1982, a Tasmanian wildlife ranger named Hans Narding woke in his vehicle in a remote swamp forest at 2 a.m., swept a torch around out of habit, and reported an adult male thylacine standing 6 or 7 meters away, 12 black stripes on a sandy coat, which opened its jaw at him before vanishing. None of these sightings has ever been confirmed, but the fact that a country keeps looking tells you what this animal means there. 90 years is nothing in extinction terms, and it changes everything, because the genetic material is in far better condition than anything else on this list. Colossal describes the Thylacine project as their most advanced and ahead of schedule. They've sequenced a complete genome, and they did something rarer than that. They recovered RNA from a preserved specimen, a 110-year-old animal that had been sitting in ethanol in a Melbourne museum. That distinction matters more than it sounds. DNA tells you which genes an animal had. RNA tells you which genes were actually switched on and which tissues doing what. Almost nobody has that for an extinct species. The base animal is the fat-tailed dunnart, the thylacine's closest living relative. And marsupials have a real advantage here. Their young are born at an almost embryonic stage and finish developing in the pouch. That means a dramatically shorter pregnancy than a placental mammal and a much easier surrogacy problem. The catch is size. A dunnart is about the size of a mouse. A thylacine was about the size of a medium dog. You are asking a mouse-sized animal to be the starting point for something roughly 100 times heavier. But there's one more thing that puts the thylacine near the top, and it isn't genetic. Tasmania still has the habitat. The forests are intact. The prey species are there. If you produced a functional thylacine tomorrow, there is somewhere for it to actually live, which is more than you can say for a mammoth or a moa. Number 2, the woolly mammoth. This is the flagship. It's the reason colossal exists. The mammoths didn't all vanish at the end of the Ice Age, which is the part most people get wrong. The mainland populations died out around 10,000 years ago, but a group got stranded on Wrangel Island off the Siberian coast when sea levels rose, and they survived there for another 6,000 years. The last woolly mammoths on Earth died around 4,000 years ago. The pyramids at Giza were already standing. There were mammoths alive while ancient Egypt was building. The reason that last population died is genuinely unresolved. The obvious theory was inbreeding collapse, a tiny isolated population accumulating fatal mutations until it couldn't produce healthy offspring anymore. Scientists called it the genomic meltdown model, and it made sense. Then, a 2024 study analyzed 21 mammoth genomes spanning 50,000 years, including 14 from Wrangel Island itself, and found the opposite of what everyone expected. Yes, the population was inbred. Yes, genetic diversity was low. But the most damaging mutations were actually being purged over time. The individuals carrying the worst variants weren't reproducing, so those variants disappeared. The population was, in the researchers' words, doing okay despite the inbreeding, which leaves a mystery. Humans didn't reach Wrangel Island until roughly 400 years after the mammoths vanished, so we didn't kill these ones. Climate change alone doesn't account for it. Something sudden did it, most likely disease, or a single bad environmental event hitting a population with almost no genetic variation in its immune genes to fall back on. That's the part conservationists point to, because it's a live warning rather than a historical footnote. A small population can look stable right up until it isn't. The decline is gradual, then there's a threshold, then it's over, and the warning usually arrives too late to act on. Here's where the project actually stands. Colossal has identified roughly 85 genes they consider relevant, and as of early this year, they'd edited about 25 of them into Asian elephant cells. They expect embryos ready for implantation by the end of 2026 and a calf born in 2028. That 2028 date is everywhere online, and it's worth knowing exactly what it is, the company's own target, not an independent projection. In March 2025, they produced their proof of concept, the woolly mouse. They took mammoth genetic information and engineered a mouse with a long golden coat and altered fat metabolism, both mammoth traits. A mouse gestates in 20 days, so you can test edits quickly. It was real science and it worked. There's also a misconception worth clearing up here, because it's the single most common thing people believe about mammoths. You cannot clone one. Not from what we have. Mammoth remains in Siberian permafrost are extraordinarily well preserved. Some specimens still have hair, skin, even stomach contents. It's the best preserved ancient material of any extinct megafauna by a wide margin. And it's still not enough, because cloning requires an intact living cell with an intact nucleus, and freezing destroys that. Ice crystals rupture cell structures. DNA breaks apart into fragments over thousands of years, regardless of how cold it is. So what we actually have is a shattered library, millions of short fragments that have to be computationally reassembled against the Asian elephant genome as a template. That reconstruction is impressive work, but it means every gap gets filled in with elephant. You're not reading the mammoth's book. You're reading an elephant's book with mammoth sentences pasted in wherever we could recover them. Scaling it up is where the problems live. To birth a mammoth, you need an elephant, and Asian elephants are endangered. Using them as surrogates for experimental pregnancies is an ethical problem with no clean answer. Elephant gestation runs about 22 months, so every failed attempt costs nearly 2 years, and cloning failure rates are ugly, as Celia's clone demonstrated. Colossal solution is one of the strangest things happening in biology right now. They're building artificial wombs. A 17-person team, and they've already used a prototype artificial uterus to culture fertilized marsupial embryos more than halfway through pregnancy. In order to bring back a mammoth, they are inventing a machine that grows animals outside of a body. But ask the honest question, if a calf is born in 2028, what will it be? An Asian elephant with somewhere between 25 and 85 edited genes. Hairy, cold tolerant, over 99% Asian elephant. That's not a mammoth walking out of the permafrost. It's an elephant wearing a mammoth's best features. Number 1, the dire wolf. And here's why it's number 1. Because it's already done. Or that's what was announced. In October 2024, 2 wolf pups were born in a lab in the United States, Romulus and Remus. A third, Khaleesi, followed in January 2025. In April 2025, Colossal announced them to the world as dire wolves, the first de-extinct animals in history. It made the cover of Time magazine. The dire wolf is not fictional, despite what most people assume from Game of Thrones. Anacion dirus was real. It lived across North America through the Ice Age, bigger and more powerfully built than a gray wolf, and it disappeared around 10 to 13,000 years ago. To build the pups, Colossal sequenced the dire wolf genome from 2 fossils, a 13,000-year-old tooth and a 72,000-year-old inner ear bone. Then they took blood cells from a living gray wolf, edited the DNA, and cloned the result into surrogate hound mixes. The animals are real and they're doing well. By 6 months, the males were nearly 4 feet long and 80 pounds, projected to reach 6 feet and 150. They began howling at around 2 weeks. They stalk, they hunt, and they keep their distance from their own handlers the way wild wolves do, rather than domestic dogs. So, is it a dire wolf? Here are the numbers. 20 edits across 14 genes. The gray wolf genome is roughly 2.447 billion base pairs. The dire wolf genome differs from it by about 12 million DNA letters. Colossal changed 20 of them. 15 of those 20 match sequences actually found in dire wolf DNA. The other 5 were designed to produce dire wolf-like traits using variants that already exist in gray wolves, including the white coat. That coat is worth a second look. Colossal's own analysis suggested pale coloration, but the gene they identified carried a risk of blindness and deafness. So they substituted a different one from domestic dogs. One of Colossal's own scientific advisors, evolutionary geneticist Tom Gilbert, said flatly that there's no chance a real dire wolf looked like that. And multiple scientists have pointed out the white coat resembles Game of Thrones more than paleontology. The response from the field was fast and unusually blunt. Philip Seddon, the zoologist who chaired the IUCN task force that wrote the actual definition of de-extinction, said these are genetically modified gray wolves, not dire wolves. A paleontologist wrote that the company can't do de-extinction, only tweak living animals to have primitive features. A geneticist called them, generously, 100,000th dire wolf. The comparison that lands hardest came from ecologist writing in The Conversation, humans and chimpanzees share about 98.8% of our genome. Would 20 edits to a chimpanzee make it a person? Here's what makes this genuinely interesting rather than a simple debunk. Colossal doesn't really disagree. Beth Shapiro is their chief science officer and one of the most respected paleogeneticists working. Her own description to New Scientist was that it isn't possible to bring back something identical to a species that used to be alive, that these animals are gray wolves with 20 edits, cloned. She said openly that her colleagues in taxonomy would say it isn't a dire wolf. Her position is that species is a human construct, a useful label, and that the goal was healthy animals carrying dire wolf traits, not a molecular replica. So both things are true simultaneously. It's a real technical milestone, The first time ancient DNA has been used to engineer traits into an animal that survived to birth and thrived. And it is not a dire wolf. That gap is what this entire list is actually about. None of these 10 projects are resurrections. What's being built are functional proxies, living animals, edited to carry some traits of dead ones. A cold-adapted elephant, a Dunnart-derived marsupial predator, a pigeon that lays a dodo-ish bird, gray wolves with 15 dire wolf variants. In almost every case, the original genome isn't coming back because we don't have it intact. Ancient DNA shatters into fragments, and what gets reconstructed is a best guess assembly with gaps. That leads to the criticism that deserves real weight, because it comes from working conservationists rather than cynics. The argument is that de-extinction sells false hope. If the public believes extinction is reversible, that a species lost today can be rebuilt later, the urgency drains out of conservation. Ecologists have made this case directly. These announcements risk minimizing the extinction crisis while pulling attention and funding away from methods that already work. One geneticist put the sharper version: instead of engineering approximations of a dead canid, that effort could go to protecting the gray wolves that are still here and still being lost. Colossal's answer is that this is a false choice, and there's substance to it. The gene editing work from the Thylacine project has been applied to the northern quoll, an endangered Australian marsupial being wiped out by invasive cane toads. They've cloned red wolves, among the most endangered canids on Earth, to add genetic diversity to a population that desperately needs it. The embryo work feeds the Rhino program. The artificial womb research may ultimately matter far more for endangered species than extinct ones. In February 2026, they opened a biovault in Dubai, a permanent facility for storing genetic material from living species. So who's right depends entirely on where this technology gets pointed. At animals that are still alive and still savable, the critics' fear doesn't materialize. At magazine covers, it does. Which brings us back to a goat in Spain. Look at what every project on this list has in common. The Thylacine is the most advanced because we have well-preserved museum specimens, because someone kept the bodies. The Rhino program exists because 2 animals are still breathing and eggs can still be harvested. Celia's clone was possible only because somebody took a skin biopsy 10 months before a tree fell on her. Every single one of these depends on material collected while the animal was still alive. That's the real lesson here, and it's the opposite of the one the headlines suggest. De-extinction doesn't mean we get a second chance. It means the window closes fast. And everything we can do afterward is limited entirely by what somebody bothered to save beforehand. Think about how thin those margins were. Celia's clone existed because a researcher took a biopsy on a routine capture 10 months before a random tree fell. The Thylacine project is the most advanced on this list because a Melbourne taxidermist happened to preserve a specimen in ethanol rather than drying it out. And nobody threw the jar away for 110 years. The rhino embryos exist because someone collected sperm from males who were still breathing at the time and had the foresight to freeze it. None of that was heroic planning. Most of it was luck or habit or a museum with enough shelf space. Which is exactly why the least glamorous thing Colossal has built might turn out to be the most important, that biovault in Dubai, a facility whose entire purpose is storing genetic material from species that are still alive right now. No headlines, no magazine covers, just a freezer and the assumption that we're going to need what's inside it. Extinction is still permanent. We've just gotten very good at building something that looks like it isn't.